Hi everyone, my name is Mia Broad and I'm third year PhD candidate in the Department of Neurology, and today I would like to talk to you about my thesis work around epilepsy. Epilepsy is a chronic neurological disorder characterized by frequent seizures or convulsions and can be due to a number of factors, including genetic factors, brain injury, or prior infection. 1 in 26 individuals in the United States will develop epilepsy in their lifetime. And currently, 3 million individuals have epilepsy in the United States, with 1 in 200 children being diagnosed. I am particularly interested in a group of epilepsy disorders called the developmental and epileptic encephalopathies, or the DEEs. And the DEEs are a group of rare and severe epilepsy disorders that commonly begin in infancy and childhood and are characterized by frequent seizures, intellectual disability, and developmental delay. About 50% of DEEs are explained by genetic cause, but we don't know the other 50%. So at Northwestern, I am studying as a computational biologist, which means I look at biological data through the lens of computer programming to better understand biological processes. But I'm also training as a human geneticist. And as a human geneticist, I study our genes. And so our genes are composed of a molecule called DNA. And DNA undergoes a biological process called transcription to become another molecule called RNA. And so this is what our genes become. RNA can also be represented like this, like an RNA transcript, which is a literal record of our DNA and is composed of these coding blocks called exons, followed by a stop sign that tells the biological machinery to stop reading an RNA transcript. And finally, RNA undergoes another biological process called translation. And translation creates proteins, which are the physical manifestation of our genes. It's our skin, it's our hair, it's everything in our cells are made up of different proteins. And this total process is called gene expression. You can kind of think of gene expression like this, where DNA is like a recipe book, and RNA is like the mise en place or the ingredients that make up a dish, and finally the proteins are the final meal. And so we have a special type of exon in our DNA called poison exons. And poison exons are actually naturally occurring. This may be the only time where something has the name poison is actually good for us. And poison exons are special because they regulate gene expression. The way that poison exons do this is that they are incorporated into an RNA at times. And this tells the RNA transcript that now the stop sign is in the middle of the transcript. So this will tell the biological machinery that reads the RNA to not create protein. You can see how this could be a good way to fine-tune and orchestrate really complex biological processes like human neurodevelopment. Poison exons are critical for normal human neurodevelopment. But we have found in our lab and in others that there can be harmful mutations near poison exons that literally make them poisonous because now they are dysregulated by these mutations and can contribute to different neurodevelopmental disorders like the DEEs. And so we now know that poison exons are important for human neurodevelopment and are also implicated in diseases, but actually they are historically understudied. Poison exons have been historically understudied for two main reasons. One, poison exons are quickly degraded. Just naturally, they are quickly degraded as soon as they are made, and so it makes it really hard to detect and study them. This is compounded by the fact that there has been limitations in previous technology to then study the gene expression of poison exons. But this is where I want to answer this call. I am developing a special computational algorithm called POISON, which stands for Poison Exon Discovery for long-read transcriptomes. And this tool will be able to systematically identify poison exons. And I'm especially interested in looking at poison exons in the context of neurodevelopment to understand their patterns and how they operate in neurodevelopment. And to do this, I will be growing brain organoids, which are tiny brains that you can grow in the lab, and using an innovative modern technology called long-read sequencing, which will let me look at poison exons in the context of neurodevelopment. And applying my tool, I will be able to generate an atlas of poison exons. And this can serve as a repository of poison exons where we now know the locations of them and their patterns in neurodevelopment. And this can help speed up the studies of poison exons in neurodevelopment as well as in other biological processes. But what I'm really interested in is how this repository of poison exons can help us create therapeutics for these DEEs. Currently, we have a drug in clinical trials called an ASO that targets poison exons and can treat a severe form of epilepsy called Dravet syndrome. And so my hope is that with a repository that I curate of poison exons, it can lead to a database of therapeutic targets to help treat these severe forms of epilepsy and hopefully advance in better treatments for children and adults suffering from epilepsy disorders. Thank you.